I'm really, really excited to have the opportunity to officially share with you and present our 2 Catalyst Research Award grantees awards this year. We don't often have the opportunity to share with all of you our research awardees, and I will just say that it was quite an extensive experience this year. We had 26 really good research grant applications, and to let all of you know, this is not a process that we undertake lightly. It is a year-long process from when we send out the request for proposals. The researchers have to put a really extensive proposal together, and those proposals are then reviewed by a group, our scientific review committee, which has experts in the field of cutaneous lymphoma that review those applications. They score them according to the National Institutes of Health research scoring process. And every proposal gets a numerical score, and then the group comes together and they have a conversation about which of the research grants are the best to be awarded. And I will say it was a really tight and very challenging decision to make. And once the scientific review committee makes the recommendations, our 2 research chairs of our board of directors, Dr. Stephen Horowitz and Dr. Yan Kim, then do a final approval, and our board of directors approves the grant. So, this is a big deal. And I'm thrilled that we as the patient community are able to support research in cutaneous lymphoma. If any of you have had the chance to read our newly published research roadmap, that was 3 years of our research advisory council coming up with that whole plan. And generally speaking, we are, in their eyes, 15 years behind the B-cell lymphoma research community as far as what we understand and know about the disease. So we have a lot of work to be done. And I think all of us in the patient community see that every day as we ask our clinicians, well, why did I get this disease? Or why isn't this treatment working anymore? Or what else is available to me to treat this disease? And there's so many questions that need to be answered. And so anyway, all that as a little preamble. Can you tell I get excited about the research? I'm just honored and thrilled to be able to invite our 2 awardees to be with us and to share a little bit about their projects. And we'll be following them throughout the course of the year and the term of the grant. So we'll also be able to share more information about their particular presentations and their projects with you as time goes along. So anyway, I would like to introduce you and welcome our 2 awardees. We have Dr. Asia Angelova, who joins us from Heidelberg, Germany. Welcome. And we have Professor Pietro Quaglino, who is joining us from Turin, Italy. So just really thank you for taking some time out of your Saturday evening to join us. And I'm going to ask Asia first to share a little bit about her study and her project. It's very exciting. She's got a really interesting question she's looking at related to how we may be able to use viruses, and she'll correct me if I get all this wrong, but how we can use viruses to attack the cancer cells. And it's really innovative and novel project in the lab to look to see if she can find a way to leverage that in treating cutaneous lymphoma. And she's a senior scientist and project leader at the German Cancer Research Center, as I said, in Heidelberg. And we'll I'll give you the formal name of her project. It is H1 parvovirus-induced oncolysis. So she's really testing my medical terminology. Anyway, and tumor microenvironment immune stimulation in a novel heterotypic spheroid model of cutaneous T-cell lymphoma. But she will be able to explain it much better in layman's terms. So I'm going to turn it over to Aisha to share a little bit about her project.
Yes, thank you very much, Susan. I first would like to say that I'm, of course, very much excited to share this conference experience with you. And I am extremely grateful to the Foundation for supporting our research proposal. And I need to say that we are happy that our ambition to test this novel approach to cutaneous lymphoma treatment was so highly appreciated. Briefly, this novel approach relies on the usage of the so-called oncolytic virus. That is a therapeutic, a good virus that is selectively killing the tumor cells while sparing the normal ones. And our division at the German Cancer Research Center in Heidelberg has over 25 years of tradition and expertise and experience in testing the anti-cancer activities of the oncolytic H1 parvovirus. We have tested the anti-cancer activities of H1 parvovirus in various tumor models. And furthermore, we have conducted 2 clinical trials, one in glioblastoma patients, in brain tumor patients, and one in pancreatic carcinoma patients. However, it has to be stressed that the potential of oncolytic virus therapy in general and of parvovirus H1 in particular has very poorly been studied or not at all studied in the context of cutaneous T-cell lymphoma. And therefore, we decided to focus our research efforts on this condition because we know that although being relatively rare and with a generally benign prognosis, it may become devastating upon systemic spread. Therefore, we aim at establishing a novel in vitro model of CTCL, and this model will reconstitute the in vivo relevant presence of a complex tumor microenvironment. And we would like to investigate in this model the anti-tumor activities of the parvovirus and also the immune stimulation of the tumor microenvironment and the triggering of anti-cancer immune responses. It is in general our hope and our ultimate goal to be able to demonstrate through these pilot preclinical studies the curative potential of parvovirus therapy, and thus to pave the way to the further development of parvovirus therapy as a novel strategy against cutaneous lymphoma. So thank you once more. Yes, I'm happy to be here with you, and I wish all of you to stay healthy and safe. Thank you.
Thank you, Dr. Angelova. That sounds so exciting, and we really look forward to learning more as your project continues and to watching how this all unfolds. So thank you so much.
Thank you.
Thank you. So Professor Quaglino, I'd like to turn it over to you. You join us from the Department of Dermatology and you're on the Faculty of Medicine and Surgery at the University of Turin in Italy, and also a very prominent and active member of the International Society of Cutaneous Lymphoma. So you also, in addition to doing your research, you treat patients every day, and you see people every day living with cutaneous lymphoma. And your project is entitled, excuse me for having to read these, but I want to get them right.
No, it's okay, it's complicated.
Yes. Mechanistic insights into the CD39, CD73 adenosinergic immunosuppressive access in patients with Sézary syndrome, association with disease course and treatment response. And if I interpret your statement a little bit, this is exciting on kind of a very different side of the research spectrum from Dr. Angelova. You're looking at, particularly in Sézary syndrome, why some treatments stop working, and what is it that makes the disease so smart that it can really dodge the treatments that we're trying to deliver to kill the cancer. That's kind of in a very high level nutshell. Hopefully I'm not way off base on that, but I think, as we all know, the challenge in the clinic with patients, particularly with Sézary, which is harder to treat over the long course of time, and sometimes the disease gets really smart, and those things that were working are now not working, and we're not always sure why. So I'm going to turn it over to you to share your work and to share with everybody what it is that you're looking to accomplish with this research project.
Yeah, many thanks, dear Susan, for this very kind introduction. And I have to say that really is a privilege for me to be with you this evening in Italy and this afternoon in US. So good afternoon to everybody. And I definitely say we would heartily thank all of you, the Cutaneous Lymphoma Foundation, you, Susan, the president, for supporting our project and make it possible to study this specific field, which, as you correctly mentioned, the main field is immune modulation and immune suppression. So try to study and understand how this patient become immune suppressed and got all these problems with infectious complications that, as you know, complicate in a very difficult way the treatment and disease course for this patient. And first, is to say that for having a good project, we have to work with friends, and Cutaneous Lymphoma Foundation is definitely a friend, but I have also a friend of mine in my department because I am a clinician, so I treat a patient, and I am an honor to work with these colleagues that work with me, and from the Laboratory of Genetics, Professor Ada Funaro and Ortolan, and from the Department of Pathology, Rebecca Senetta, who collaborate with me in working in this project. So mainly, as I told you before, sorry, try to move. Okay, perfect. Okay. The main field is immune modulation, and we focus our attention on this molecule, which is adenosine, which is a small molecule, is an immunosuppressive metabolite in the tumor microenvironment. And we try to get some more insight in the activity of this molecule in the immune system of this patient, how it modulates, and which is the impact on the immune system homeostasis, and thus to reflect on clinical management of these patients. And so we try to study the production of adenosine and also the pathway through which adenosine is produced by the cells. So there is a canonical pathway mediated through CD39 and CD73, and an alternative pathway mediated by CD38. So basically, we studied these pathways using flow cytometry, so blood evaluation of phenotypes of blood circulating cells, and these are the molecules that we study related to this pathway. And also we studied immunohistochemical expression of these molecules, so in tissues. And then we will also try to make some experiment about production by malignant tumor cells isolated from Sézary patients of this metabolite and the evaluation of the potential of adenosine to modulate immune system in vitro. And we will try to study this expression in 2 cohorts of patients. One prospective cohort where this cohort will be analyzed by flow, immuno, and lab analysis, and a retrospective cohort for controls also. And this cohort as retrospective will be studied only with immunohistochemistry on tissue specimens. Just to summarize our aims, for sure to try to translate this evidence of expression molecules and laboratory for the clinical management of these patients. So try to identify an immunophenotypic signature defining specific subset of patients with different levels of immune suppression, and to correlate adenosine expression pathway related with the clinical features, thus response to treatment, response, duration, and development of infection complications. And last but not least, try to evaluate the ability of specific inhibitors of this pathway, if relevant, in order to try to restore before in vitro and then hopefully in the clinical management in order to restore the anti-tumor immune response and so revert immune suppression. I think that's all. So many thanks again. These are some pictures of my town just to show you that Turin is not only hospital and cutaneous lymphomas, but it's also monument, some fun also, even if the time now is really difficult, but we are seeing the light and the situation is improving, and I hope also in US things are becoming better, and we do hope to be over this awful period as soon as possible. So I hope to see all of you in Turin for sure, but I hope also to see all of you in presence and net meetings. And thank you so much, and I think that's all.
Great, thank you so much. I know for folks that may not be super scientific, it's a lot of terminology, but I think it also points to the importance of this kind of research and the broad stretch of research that is really required, that the things we need to know and understand to create new treatments, but then also to understand how we can better utilize current treatments, and diving into more and more of that specific approach for the patient. The more we can understand about their particular iteration of the disease, the more we know, the better we can treat it. And then what Dr. Angelova is doing is really about, can we find new ways, new things that could work to kill the cancer? Right, that so these different combinations are all required, and you know, this is
we also put together,
right? Yes,
because
and
we need more.
Yeah, and this is the tip of the iceberg, everybody, of like, when I have the privilege of sitting in the scientific meetings and listening to everybody, and you know, there's so much more that we need to do, and there's all this work being done. And I think for us as the patient community, the more that we are able to raise the funds to be able to support the research, the more, the faster we can move forward to better manage everybody and find new ways of treating. So this is a beautiful step in the right direction. Thank you both so much for sharing your work with us.
Definitely our best and work hard for this project.
Yes, thank you, and we'll be sharing some lay words that both of the researchers have put together, so you can read a little bit more about their projects, and then as they finish up, we'll have some reports we can share with you as well. So we'll hope to have you both back after you have a little bit more information to share with us. Yeah,
yes, for sure.
Fabulous. Well, thank you. Have a lovely rest of your Saturday evening. Thanks for spending a
little
bit
of it. Have a nice afternoon.
Thank you. Greetings from Germany.
Yes.
Ciao, 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 ciao from Italy.
Have a nice day. Thank you.
Bye bye.
You too. Thank you.
Bye bye. Thank you again.
Okay.
Bye.
Bye-bye.
Bye bye.